Well, welcome everybody. Thank you so much for being here. This is actually our fourth C-MAIKI Symposium. Each one's taken a little bit of a different tune, but I'm very excited to welcome you here today. So thank you for taking the time. I know it's a very busy time at the end of the semester, so it's great to see so many familiar faces and some new ones as well. I'm Nicole Hynson. I'm the director of C-MAIKI. Just a little bit of housekeeping. If you're looking for the bathrooms, they are downstairs out and to the right. You'll see a foosball table, and then you'll know that you're close to the bathrooms. All right. So first off, I just want to give some acknowledgements. I want to send my deepest gratitude to the UH Foundation. Not only did they help us secure this nice space for our symposium today, but they are the ones that have supported much of the work that you're going to be hearing about today. So thank you very much to the UH Foundation. We have Tim Dolan here and others from the foundation. So thank you too for taking the time out of your day to attend this. I also want to thank the Office of Vice Provost for Research and Scholarship, under which our center sits. And we have Rob Wright here today. So thank you for taking the time to join us. And then I would also like to thank the Walter Dodds Junior Rye Center, so where we are today and having this nice space of the PACE classroom. And then I'd be remiss not to give like a deep thank you, and how about a round of applause for Nicole Yoniishi. So all the beautiful swag and program and food and everything that you're seeing today is largely because of Nicole's work, so thank you. And then also because of our two faculty committee volunteer members, Dr. Maggie Yuan and Dr. Alex Kelli, who have been helpful throughout this process of planning. All right, so with that, I just have a few words, and we'll see how this lands with you guys, but I just want to pose this question to the group. All right, so my question today is, why have a microbiome center, right? We're sitting in this time that's very challenging when it comes to the federal landscape for science funding. This is a brand new headline that just came out, right? Slasher sequel, Trump again proposes major cuts to US science spending, but we all know it doesn't end there, right? So we've seen that the whole board of the National Science Foundation was just fired. There's been holdups from many federal agencies with funding, NOAA, USDA, the NIH. And perhaps we could say, well, you know, because of the way that our system works, there are some checks and balances in place, hopefully, right? So we see lines like this: US lawmakers vote to reject Trump's budget and then hopefully restore some of our funding that we rely on to do the work. So how is it that we can justify our existence, and how can we find the motivation for the existence of such a center as ours during this really challenging time? And that's what I want to talk about a little bit. So we could say, right, that this is just a practical endeavor. And perhaps this would fit very well within some of the things that we're hearing about under like DOGE, right? So it's just practical. We reduce redundancy, we centralize resources by having a center, and we increase efficiency. So how do we do that, right? So we do that through mechanisms such as the MGAL. So this is our genomic center where all of us can send our samples and we can have them run to get DNA sequence data. And we don't have to recapitulate this in our own units. So it's efficient and it reduces redundancy, right? So maybe this is it. Maybe this is just the reason. But I would actually like to argue that it's these intangibles that are really critical and why we need our center. And I'm putting these intangibles under two broad categories. One is transdisciplinary synergies, and the other is community and intellectual home. So I'm going to show you a couple products of having these more intangibles. However, I just want to iterate that it's not so much the products, but it's the fact that these two things are what really bring us together to continue the good work that we're doing. But for example, something like this work, which is now came out in TNAS a few years ago, this is an example of transdisciplinary synergies. So this was the first of its kind, complete census of an entire ecosystem's microbiome. And this would not have been possible had we not had expertise from oceans, freshwater, land, hosts, all working here together at UH Manoa. So this is an example of a product from a transdisciplinary synergy. And then the next one, it might seem kind of like the beginning of a joke. So I'm going to say, you know, a mathematician and a soil microbiologist walk into a bar. But actually, what I'm talking about here is a collaboration between Professor New Win from Tropical Plant and Soil Science and Professor Daisuke Takagi from the Mathematics Department that came about from interactions with C-MAIKI and led to this really nice award from the NIH on bacterial dispersal and nutrient transport on fungal highways. So very cool, very cutting edge work that wouldn't have, I don't think that this relationship would have formed in the absence of the center. But perhaps like the better way to think about it is, what is it that brought us all together in the first place? And it was the love of microbes, right? We all love microbes. Woo, microbes, right? And I'd like to remind, especially the younger folks in the room, right, that as recently as 2008, this is what our tree of life looked like, where the vast majority of what you'd see are non-microbial organisms. And then microbes are inhabiting this little tiny, like, what is that even? Like they're down there in the roots or something? Who knows? What is that? But we know now, right, that the diversity of life is overwhelmingly microbial, and that plants and animals comprise just this tiny little branch. So perhaps not only does this bring us together, but it has fundamentally changed our worldview, right? And I would like to call this a movement from a eukaryotic-centric worldview to a microcentric worldview. And I'm going to share this quote that I shared with some of you previously, but I just love it. So this is from this article, Love is a Microbe Too, where we say microbiome research can be envisioned as a narcissistic offense. Scientific breakthroughs, Freud argues, challenge our self-image as human beings. After discovering that we are not the masters of our universe, and this was Copernicus, of course, nor the masters of creation, Darwin, right? We now discover that we are not even masters in our own house. Microbes threaten to undermine our sense of agency and self-mastery. So what does this mean? Does this mean that we should all run into a corner and hide and wait for the next pandemic? No, I would say the opposite. I would say instead what it's telling us is that could it be then that microbiomes actually have something to teach us? And I would say indeed they do. And I'd like to categorize some of their primary lessons into a few different areas. Oftentimes in society, we think of redundancy as superfluous. I think you can look to the microbiome to see that redundancy can actually mean resiliency. So having expertise that could be seen as redundant can actually lead to a resilient system. Complexity is oftentimes, again, seen in our society as something to do away with, to simplify, to be reductionist. But I would argue that the microbiome tells us that complexity can actually engender efficiency. So it's okay to be redundant. It's okay to be complex. And then finally, I think we can all agree that synergies can enhance outcomes, and that microbiomes are a great example of this. So why should we be any different? And I'd just like to kind of round out with saying, I encourage all of you to be expansive in the work that you do, to be expansive in the relationships that you form. So even in this time of sharp and pointed reductions led primarily just by the politics in our country, we should not narrow the ways of thinking. The data do not care who is in the White House, nor do the microbes, and really strengthen numbers. So this to me is the justification for our center. And I'm really proud today to introduce our speakers. because our speakers and poster presenters are really the leaders in our center and the leaders of tomorrow. Justin Berg, PhD student in marine biology, and Kelli Konicek, who's a PhD student in zoology, have both been supported in part by the UH Foundation. And then we ran for the second time this competition that we call Mahi Microbe, where we solicited applications from students and postdocs, asking them to identify a clear and pressing Hawaii-based problem and articulate how their research in microbiome science could actually help solve this problem and have measurable and translational impacts. We ran this competition, and I just would like to give a shout out to our volunteer judges who were both community members from our University of Hawaii at Manoa, Ohana, as well as some of our community members from outside of the university working in the nonprofit space. And our three additional student speakers today are Stephanie Murray, who's a PhD student in computer science, Sebastian Church, who's a PhD student in tropical plant and soil science, and Mamo Waianuhea, who's a PhD student in zoology. And these are our winners of the Mahi Microbe competition. So the other thing I'd just like to point out when you're looking at this slide is notice how many different units across the University of Hawaii are represented here. And that's another thing about the center, is that it brings together these people from all these different parts of our ecosystem that would not normally interact. So I'll just close with this sentiment: be like a microbe. Be bold, traverse boundaries. So some microbes don't care whether they're in the soil, the water, or host. Traverse those boundaries, whether it's introducing yourself to someone new at this meeting today, or perhaps thinking about a new method that you could use in your own work, attending a new meeting. Be adaptable, thrive in even challenging conditions. Microbes on a daily basis face some of our most challenging conditions on the planet and can thrive. There's no reason that any of us should necessarily be any different. So thrive under challenges. Network, the whole is greater than the sums. Make those connections here and in your life outside of this room. And be resilient. Tough times call for new innovations, finding new ways of doing things, not just throwing up our hands and saying we cannot. And with that, I'd just like to say thank you. And it's really a pleasure and an honor to introduce Jack Gilbert, who's here today with us from UC San Diego. And I have a little blurb about Jack that I'm going to read. So please bear with me while I go through this, because it's really just impressive and I love it. So he co-founded the Earth Microbiome Project and the American Gut Project. He's the founding editor-in-chief of M Systems Journal and has authored over 450 peer-reviewed publications and book chapters, and has co-authored Dirt is Good, a popular science guide to the microbiome in children's health. In 2023, he became the president of Applied Microbiology International and won the 2023 IFF Microbiome Science Prize. In 2025, he was elected to the American Academy of Microbiology and became the co-chair for the new IUCN Microbial Conservation Specialist Group. And I really encourage you to look him up online because there's just some amazing interviews and podcasts. My favorite one is the one with Ed Young. I love that. And so if you don't know Ed Young, he's a science writer who wrote the book I Contain Multitudes, which I highly recommend you pick up a copy. And he has one with Jack called A World Without Microbes. So if you've ever struggled with how to tell your grandma that microbes matter, just sit down and watch this video. Today, he's going to be speaking with us about a topic of particular relevance to our island home, sustainability and the role that microbiome innovation can play in a more sustainable future worldwide. So please help me in welcoming Dr. Gilbert.
Thank you. I opted for the mic for karaoke, so hopefully this is good. Oh, and I get a necklace like that. Thank you. I'm a microbiome scientist, and she was just asking me, what's your name again? Mamo. Mamo was just asking me what kind of thing I specialize in. My ADHD does not allow me to specialize in anything. I literally am the jack of all trades, master of none. So I will jump around subjects and I will go on weird little tangents, and I apologize in advance. And he has to hit me with things if I go over line. Okay. Everything I'm going to talk about is pushing towards translation, getting things out into the market. And the reason for that is because we've run out of time, not just in this talk, but generally. We need options, we need solutions that we can use, deploy, test, validate in the field, alongside doing some of the basic research, which is so fundamental to us being scientists. So for me, the key precept of this entire talk is we need to get things out there. We need to be engaged with industry. We need to be engaged with policy, and we need to be making a difference now, not in 5 years' time. Okay. And with that in mind, I do a lot of work with companies, and some of the work that I do is represented here. I currently sit on the scientific advisory board, and I'm chief scientist. I don't really know what that is because it's not an executive position, for a company called Oath Inc., which is a public benefit corporation, and you can look them up. Okay, so I'm also president of one of the oldest microbiology societies in the world, but it's the oldest in the UK, called Applied Microbiology International, used to be called the Society for Applied Microbiology. And this is an organization that really does pin its hopes on applied science. Use-inspired science is another great term for that. The opportunity to take the technologies that we're developing and deploy them at scale, and test them at scale, so that we can have an impact upon the problems, solving those greatest challenges with microbiology. And I think, you're saying Nicole's pretty inspiring. It's hard to follow that pitch, but, that's the key piece. Microbiology has overcome a lot of things. It has a lot of solutions embedded in that one phrase. And if we can just harness those solutions, we can play a significant role in shaping the sustainability and productivity of our ecosystems and environment for the survival of our species. And I will say that that's still important, right? Our brains are weird. We're very egocentric, but none of us would be in this room, and none of us would give a crap about a coral reef if it wasn't for the fact that we do, right? I know that's axiomatic, but we do care about it. And that's the reason we need to do something about it, not because of its necessary intrinsic value, but because it's valuable to us. And if you don't want to argue that point, that's fine. But it is important to us. So let's take a role in playing and helping out that paradigm. There are 17 Sustainable Development Goals to the UN. If you do not know what the Sustainable Development Goals are, please look them up. They're a really, really powerful way of structuring your scientific questions, putting your discipline into action, because they have been developed and validated extensively at an international level. And you can look this up every single year. A new report comes out saying how we, as a community of human beings on this one planet that we call home, have managed to achieve any of these components. And my hope is that we can integrate microbiology into that paradigm, so that every single one of these goals has a potential microbial solution that could be part of the toolkit that we use to address them. For some of them, it already exists. So the concept of integrating microbiology into these frameworks has really already been laid out pretty well. So, for example, creating dynamic task forces that are responsible for integrating microbiology into the UN Sustainable Development Goals has already started to happen. We've also started to get local ambassadors and create a decentralized strategy, so that local communities and indigenous cultures and people living in different environments can play a role in managing their populations and their local ecosystems. Pilot testing and scalability, identifying the strategic potential of a solution, and then testing and innovating, going through a virtuous cycle of improvement so that we get those technologies out there. And then interdisciplinary collaboration, like we heard, right? You're stronger together. We're stronger building networks together. And that's why I fully agree centers of thought are incredibly valuable in helping us to achieve that outcome. And then milestones and metrics and regulatory agility and ethical considerations are the central pins that enable us as an organization to be able to facilitate the goals that we want to see deployed in the world. It's all very well saying, here's a technology. It's a much more complicated and difficult process to ensure that you've addressed all the ethical considerations for the deployment of that technology. And this is the reason nearly all of the science I'm talked to today has a social science component. And a lot of people might not consider social science a science, but it is a science. It's got science in the title. It's a really important piece because everything that we do is human, right? Whether you're studying a coral reef and you're the greenest of the green people living out in the green trees, you're still a human being and you approach it with a very humanocentric attitude. So that is social, cultural initiatives. And social science is the science of trying to understand how those social cultural initiatives work and how they can be implemented and deployed to facilitate outcomes. Okay, so let's take a good example, healthy land. Land has a wide variety of uses, agricultural, residential, industrial, recreational, and these could be really important paradigms. Microbes can play a role in all of these components, both in terms of symbiotic relationships with plants, in terms of managing the quantity and quality of soil, but also in terms of managing the health of all of the organisms that exist and interact with that space. Human land use has led to the exhaustion of nutrients in soils and contamination of land and reduction in biodiversity. This is very well understood. It was understood in 1930 in America, and a lot of work went into trying to fix that problem. And then by 1945, we completely ignored it again. Humans. Microbes can be used to increase crop production, remediate pollution, increase carbon accumulation, and improve resiliency, like we heard from Nicole. So this all can be used in terms of addressing number 15 of the UN Sustainable Development Goals, life on land. And that's a really big topic, but it's an incredibly important topic for us as a species. And some of the work we did recently went into trying to mold social science, political science, and the microbiome and plant technology science together into a unifying modeling framework to facilitate us being able to predict the potential implication of adopting novel strategies for agricultural development and deploying them at scale. Why is this important? Well, if you want to deploy a technology and you want to see and predict its impact upon the world, you better be sure that your predictions are accurate, right? We were able to demonstrate that by developing strategies to understand how technologies could be deployed and then modeling their deployment through existing social, cultural, and political frameworks, we can determine that deploying enhanced crops with microbial bioinoculants can sequester or could sequester up to 1.2 gigatons of CO2 per year. That's 7 times more than all of the existing CO2 offset offerings for the planet currently existing. So the ability to take these technologies and deploy them does exist. The frameworks and the roadmaps do exist. We can predict their potential implication. The only thing we're lacking is will. Right? We can do this if we actually have the willpower to do it. A long time ago, in a galaxy far, far away, we deployed something called the Earth Microbiome Project, which was a fun little study that we thought would be small and kind of kooky and turned into a monster. But the premise was, after about a decade, we developed an approach that allowed us to survey over 27,000, 28,000 samples around the world. I think we're now up to about 400,000 or 500,000. Over 500 scientists collaborating, generating tens of papers. It's now thousands of manuscripts, but covering, most importantly, the range of microbial existence on the planet. The idea that we can go from extremes of oxidation to reduction, extremes of temperature, and extremes of environmental stress and resilience, and try and map and track what's going on in those ecosystems. The outcome of that was maps and identifiers that helped us to understand microbial dynamics across scales. One of those, for example, which is pretty obvious and we already knew, was that soils and soil-like complex environments are extremely hyperdiverse. They have the most microbial diversity. And if you go into non-free living, so host-associated systems that are associated with soils, like the roots of plants, they are also extremely diverse. That is a hyper-diverse environment. Very complicated, but very cool to work in, especially if it has one of your favorite crops growing inside it. And we, after I get back from this, I do have to go to Sonoma to try and get donors over there to support viticulture microbiome research. And I've been trying to do that for 15, 20 years now. Somebody one day will give me money to work in vineyards. But this is cool, right? So you can take this microbiome dynamics and you can try and understand and explore how it functions. But the thing that I was most excited about is what I call microbial osmosis. The idea that microbial dynamics are extremely diverse, but things are constantly spreading from areas of hyperdiversity. If you've got an area that's hyperdiverse, an area that's low diversity, we'll nearly find nearly all of the phylogenetic diversity present in that low diversity environment is found and originating in the hyperdiverse environment. So diversity osmosis, if you will. And this comes down to the fact that microbial dynamics are hyperdiverse, and there's also hyperlocalized based upon local evolutionary processes. Every single one of these columns, and they're very, very thin columns, so remember that. Every single one of these columns is a genus on the top and a strain on the bottom. So let's focus on the genera at the top. I've colored the genera proportionally by where they're found in the world. And we can see, for example, some of them are, as Nicole was pointing out, some of them are incredibly well distributed. So they can be found in soils, sediments, they can be found in freshwater and marine systems, they can be found host-associated, non-host associated. So these genera, for example, are found across pretty much the entire breadth of environmental systems that are available to microbes to colonize. But if you go down to the strain level, i.e. the individual genotype, we get hyper specialization. So these organisms, for example, are only found in plant roots and the soil directly surrounding those roots. So that's an incredibly important thing. And we've identified individual strains that are really important, like ʻŌhiʻa. So when I was asked what my favorite microbe is and put it on the board, I wrote ʻŌhiʻa. This is a species of a single strain of Eruco microbia that is found hyper associated with the roots of particular grass species around the world. It's distributed in every one of these root systems, but it's incredibly well associated only in that environment, doesn't survive outside of it. Also, fun side fact, Escherichia coli, there are like now 70,000 genomes of Escherichia E. coli available out there. If you take some of those genomes, they can have up to 40% coding genetic diversity differences compared to other ones. If I took you and I swapped out 40% of your coding region, you could become a fungus. So like highlighting the fact that the eukaryotes are really not very diverse. Microbes can be incredibly diverse inside one species, more diverse than pretty much the entire kingdom of eukarya. So this begs the question, what is a species? But I'm not going to go down that path. So strain level variation can be extremely important, and it can be valuable because it can mean that there's endemism, and that will become important later. There's endemism in unique environments, and that endemism is a product of thousands to millions to billions of years of evolution that has supported the diverse distribution of that endemism and the biodiversity of the genetic capabilities. And we need to potentially conserve that, as I'll come to. Soils are incredibly important, but they've been under monstrous numbers of threats. Agricultural technology has led to an increase in food production. No one can say that that's not true. The Green Revolution really did work in terms of helping us to feed the world. We could have done it in other ways, but we didn't. We did it in this way, but it has had negative impacts upon the soils and the environments. In many countries, intensive crop production has badly depleted the soil and led to significant erosion of that soil. Soil health and its facility have a direct influence on the nutrient content of food crops, and also on the integrity of the soil system, meaning that it will be more resilient in the face of impending climate change outcomes. These monstrous number of impacts are manifold and incredibly important. So, for example, increased temperature can drive down soil organic carbon content in a soil based upon driving shifts in microbial metabolism. Elevated carbon dioxide in the atmosphere can actually increase it and increase biodiversity, but select for only certain species of bacteria. Increased precipitation can select for certain organisms in the soil. And if the soil is too weak, it can just lead to a funnel through of water. We need a sponge, not a funnel. Drought, permafrost thaw, fire. Fire is an incredibly damaging system. It damages the top 5 to 10 centimeters, sometimes 20 centimeters of soil, and makes it essentially barren for a period of time. And then therefore a need to drive back increases in microbial dynamics. These are all important drivers. So how can we produce enough food for the planet while building soil resilience, improving human health, and maintaining planetary biodiversity. Seems like a big thing to achieve. This is a cartoon that's on the fridge in our lab. Microbes are the littlest farmhands, and we believe that they could be extremely important if only we understand how they work and if we understand how to manipulate and how to leverage them. And I know that sounds, mercantile, but it's important that we do figure this out, because theoretically, we can alleviate a lot of those historical problems and make the system more resilient to those perturbations if we leverage these organisms. So we have developed at UCSD now another center. I love centers. The Soil Health Center, which is led by Professor Sarah Allard, that's focused explicitly on trying to understand how we can integrate those multidisciplinarities between us in order to achieve particular outcomes. So everything from engineering, biology, sociocultural, ethical, economic studies, chemistry, and physics. Of course, we don't really care about the physics, but we have to make sure that they feel included. But everything else is vitally important. And actually, the physics is super cool, right? The soil is a massive hydrology system, and understanding flow dynamics in that hydrology system has proven to be super interesting. Again, I get really pulled away by different topics, but if you give me a book on hydrology, I will read it, and it can be incredibly cool. Anyway, so we've been deploying this mechanism to explore outcomes. We've also created something called the Soil Stars. The Soil Stars is led by Professor Janet Jansen, who's one of my old friends, and she helped co-found the Earth Microbiome project with me. She's now retired, but so we gave her a job, and her job is to lead the Soil Stars. And the Soil Stars are a group of people who have come together to try and promote improvements in soil health. So we have a bunch of scientists, but we also have movie stars and rock stars in there. I know it's kind of weird, and we've got a whole bunch of politicians now, even a union leader, the Farmer Soil Union representative for the United Kingdom, for example. Groups of people in the Chinese Communist government are part of the Soil Stars. It's become weird, but it's a vehicle for allowing us to communicate with the outside world, so that we as scientists don't become super insular and say, we got the technology, just give us the money and we'll spend 25 years trying to figure out how it works. No, give us the money and we'll deploy it. We'll figure out how it works while we're testing it with farmers and with growers and with our food chain suppliers and with our politicians and with politicians on both sides of the equation, so that we can play a fundamental role in building that resiliency back into our society and our culture. And that, to me, is the most important thing that we can do through these kinds of mechanisms. It's bridge the gap. It's make sure that people don't, feel like we're living in some kind of isolated, golden chalice, that we are actually out there getting our hands dirty in the fields, working with people to facilitate real change. And so recently, literally a month and a half ago, we all met up in a place in England called the Lost Gardens of Heligan, which is run by one of our Soil Stars, which is an old farming system where the farmers used very, very regenerative practices in the 17th, 18th, and 19th centuries to build food systems that were highly sustainable. And they still run that farm the way they have done now for 400 years. And it was always considered kooky. It still is a little bit, but it's also proven to be, like many indigenous agricultural practices, incredibly functional, works, and highly efficient. So WTF, why did we not adopt it, right? But anyway, we met up down there and we wanted to define global problems that we could solve, prizes, if you will. Thank you. That would facilitate us being active in this environment and leveraging our resources to drive forward change. These ambitious, measurable, time-bound soil health missions needed to be defined. Clear performance thresholds and success metrics needed to be developed, and they needed to be able to mobilize global teams of multiple groups and open and available to everybody. So the Heligan protocols were created, and they're soon to be published, as the operating system for global soil recovery. And these may sound like super highfalutin goals, but if you don't have a highfalutin goal, trust me, you can't get people excited. And can't name drop, but weirdly, we've now got people like the Pope getting involved, and we want to make the Pope a soil star. And King Charles III, is it? Who wants to also get involved. We're going to make him a soil star because, you know, it's king. And we're going to see if we can deploy these techniques by leveraging their convening power to make real change across multiple nations. The Catholic Church is, you know, for better or worse, still an incredible landholder, and they have huge farming practices. So if we can get them to adopt regenerative practices and make it a spiritual thing, even as a die-in-the-wall atheist, hell, I will do whatever's needed to save this planet, right? And I will work with whoever needs to be worked with to make it happen, including industry. So we're built, for example, we can create a prize which anybody in the world could apply to create a universal soil blood test. Imagine just being able to create a blood test, just like I just had, which said I was pre-diabetic because I had a can of Coke 30 minutes beforehand, and apparently that wasn't a good idea. But these tests we can use to define our health and give ourselves outcomes. Let's do that for the soil. Create a short-lived carbon in soil into durable carbon pools using microbial metabolism and tailored approaches to drive that metabolic engine towards one particular outcome. For example, we could create 5 carbon lock molecules that could define a pool and the metabolic pathways that could be activated through probiotics or prebiotics to achieve those outcomes. If we put in the language that people understand, we can galvanize the global community to achieve these outcomes. And it does work. Microbial biostimulants, for example, this is data from one of our company programs. These are multiple studies, 35 studies, 9 studies here, 2 studies there, 3 studies here, 5 studies here, which show that microbial technologies, probiotics in farming systems can improve yield, biomass, soil organic carbon, bulk density, and increase water conservation above and beyond what's available using standard practices. And we're now demonstrating that we can actually significantly reduce, for example, water use in a particular system and still get the same kind of yields, if not better yields, than we would do normally. And this is a program run with the government of Rwanda across different programs. We just submitted this a few weeks ago. It's not peer-reviewed yet. But where we've taken a probiotic formulation, 3 different ones, and deployed them into systems that are using 70% of the total water they would normally use, and we get no yield penalty, a significant improvement based upon a 70% system control, but no yield penalty by adding in these bacterial probiotics back into the system. This means a farmer can save water. Farmer can therefore make a greater profit. There can be more economic engine potential for that local community, and that local community can be now self-empowered. As 65% of the farmers in this region farm an area of only 5 acres, and 87% of those farmers are women, we're talking about an engine which can support global liberation in this particular environment. We can also do it for resolving systems. So this is a mangrove. If we take a mangrove and we just try and plant trees, most of them die, especially if the mangrove system has been polluted. We did this with plant growth promoting organisms, oil remediating organisms, thrown in a bunch of other species in there, and created a probiotic matrix that allowed those trees to thrive. And so I'm going to jump through this because otherwise he's going to shout at me. But AI can be valuable. And I had to say this, right? Everyone has to use AI. It's important. But AI can be valuable. And I'd suggest go and look at this paper because we spent a long time trying to make it very simple for people to identify what AI is. But for example, we can use it for driving design. So identifying foundation models which combine diverse data and then link those microbial traits to ecosystem functions. And then, for example, allowing us to identify root microbes that produce carbon stabilizing polysaccharides, or create a synthetic community to deploy to ensure environmental capacity or compatibility. And then delivery, we can use AI optimized models to analyze soil climate and satellite data to predict the success of deploying a microbial technology. And you can even have AI-driven robotic implementation, which is happening right now for the rejuvenation of mangrove systems, that allows us to monitor how those trees are growing and deploy microbial biostimulants in order to improve their outcomes. And then assessing it. There's a ton of data being generated by these systems in an automated fashion. If we can leverage machine learning approaches, we can refine microbial interventions using continuous data feedback and integrate microbiome data into Earth system models. These are things that are happening now. They just need to be scaled up. So we can't ignore the AI train. We need to get on that train and own it. And most of us in this room have been using AI for a very long period of time. So it shouldn't be that hard for us to actually talk to the Googles and the OpenAIs and the teams of the world and say, hey, our technologies can be and our data systems can be integrated into this platform. Please help us. Microbial solutions do exist. Why are they not being deployed? Humans suck, man. How do you make humans care about something which doesn't really affect a human? You make it all about them. So does healthy soil make healthy humans healthy? How long have I got? 3 minutes. Yes, it does. We worked with the Amish and Hutterites in the Midwest. The Hutterites have very low rates of asthma. The Hutterites have very high rates of asthma. A quarter of their population has asthma. The Amish have very low rates of asthma. The only thing that really divides them is that in the Hutterite community, kids aren't allowed on the farm. You have to be a boy over the age of 14 to be allowed in the farming environment. The Amish kids, boys and girls, are living, working, and breathing in that farming environment. We wanted to see if the bacteria and the microbial signals in the soil could be useful. So we snuck into the kids' bedrooms and looked into, and we sucked up dust using little power vacuums around their bedrooms while they were sleeping, like a reverse Santa Claus or a Grinch, and took all of that dust and we gave it to mice that were had an allergic reaction to a protein called albumin. And when we give Amish dust to these mice, we reverse their eosinophilic allergic reaction, and we promote a significant increase in neutrophils. Basically, Amish dust can be a very powerful adjuvant for reversing these outcomes. Weirdly, the Amish started to try and sell their dust from the sides of the farming environment in Indiana, and I told them that they could do that, but I've got no idea the dosing, et cetera, and it'd probably be illegal. But it could be pretty cool, right? This is all about exposure. Another group has shown that if we can't bring the people to the soil, like make everybody live on a farm, maybe we could bring the soil to the people. So in this wonderful study in Finland, they took forest soil, and I hear the forests in Finland are very clean. And they took the forest soil, if anyone doesn't remember that from the first Trump presidency, come and talk to me later. You took forest soil and compared it and randomized children to either be able to play in forest soil or play in urban soil. The kids that played in forest soil had a significant reduction in inflammation and, interestingly, an improvement in school readiness scores when they went to school in kindergarten at the age of 5. This is a potential indication that there's a relationship there between healthy soil and healthy life. And we are currently running a study in Washington where we randomize kids to either receive a forest education or a urban education. And in that study, our preliminary data suggests the same thing. Kids getting the forest education have lower levels of inflammation, higher school readiness scores, and lower levels of the potential to develop obesity by the age of 5 or 6. So there's a relationship there. Kids won't remember their best day of YouTube, but they will remember being out in the soil. So I know that I don't have to preach that narrative here, but it's super important that we get kids outside more often. And I will jump over this, even though it's really freaking cool. We've been creating microbial, we're taking that soil system and sticking it into 3D printed materials and building out these incredible matrices of these 3D printed materials. If you physically interact with this surface, you will have lower inflammation in your skin. No joke. And we're building it into art that we can put out on the outside that makes people want to tactilely interact with it. We think it's important that we create architectural designs that will make people healthier. And as Nicole wanted me to say, we are trying to save the world as well through conservation. So we just formed the International Union for the Conservation of Nature's first microbial species survival commission program, the Microbiome Conservation Specialist Group. It's in our first year. We found the IUCN is the most bureaucratic system on the entire planet, but we are wading through the quagmire to try and deliver effective conservation management practice developments for microbial systems. And one of those has to be Hawaii. And your PNES paper is a key example of why we need to do these kinds of analyses. And I've run out of time, but we do have a vault in a nefarious place, which I can't talk about, where all the microbes are being held. If you're interested, go to microbiotavault.org. And that's it.
Wonderful. Okay. Yes. Hello. My name's Kelli Konicek. I'm a second-year PhD student in Joanne Yu's lab. Hi, Joanne. And I am delighted to be able to talk to you folks today about my very favorite group in the world, Hawaiian Drosophila. So the work that I'm trying to start is exploring Hawaiian Drosophila Ovi-Stimulants, which essentially means like things that can get these flies to lay eggs in specialist and generalist flies. So we'll talk a little bit about all of that in the title soon. Here's one of my tiny children that we'll talk about. Okay, but first, I would really like to acknowledge all of my funding sources because I would not be able to be up here without them. For the first year of my PhD, I had a graduate assistantship with the Oahu Army Natural Resources Program that allowed me to grab a ton of different plant material that I'll talk about later. And for my second year, I received funding, as Dr. Hynson mentioned, from the University of Hawaii Foundation, which has been just a huge blessing to be able to focus on my research. So I thank both of these organizations for like kind of giving me the kuleana to be able to work on this group. So I thank you very much. And I also thank a lot of the microbial support that I've had for this project from C-MAIKI and from some of our core programs. So thank you guys very much for allowing me to do the following research. Okay, I will introduce you to the wonderful world of Hawaiian Drosophila. Yay! I have been working with this group since 2020 as a technician in the state for the Department of Land and Natural Resources. And I really just fell in love with this group. I wanted to show you these pictures here because you can really see kind of the beautiful wing morphologies for a lot of these. And the interesting thing is that these have all descended from a single continental ancestor that was here 25 million years ago. So Hawaiian Drosophila are the largest evolutionary radiation of Drosophilids in the entire world. And it's one of the largest evolutionary radiations in the state of Hawaii, which is saying something. And the pictures here that you're seeing are really important because they're showing some of our endangered Hawaiian Drosophila. Currently, we have 14, which after talking about the trying to get endangered species listed, it's quite difficult. So the fact we've been able to get 14 is great. But lots of different species out of the gosh, it could be 1,000. It's probably around 800. A lot of those are declining, obviously, even while I've been here for the past 6 years. So there's quite a conservation need for those. One thing that I'm really interested in that I kind of want to zero on about what makes these flies special is that they lay eggs in our native plants. So they have really intimate and interesting relationships with a lot of the endemic species that also have grown up here over millions of years. And so if you can look up at this evolutionary tree here, it's color-coded based on what host plants flies lay in. And so the takeaway from that is just that there's a lot of different native families that flies lay eggs in. And there's not like a clear pattern for those, which we can talk about a little bit later. And one of the reasons that I kind of want to tell you guys about is while I was working for the state, we did the first ever reintroduction program for Hawaiian Drosophila. And so I was able to release some endangered flies out into an area that had been restored previously. And I caught this video that hopefully will play. Yes. Okay. So this is an endangered Drosophila hemipisa laying in an endangered Cyanea grimsiana, which had both been outplanted in this area. So I don't know, something about being able to see this interaction happen from our partners at the DLNR with the Plant Extinction Prevention Program and me. There's something really amazing to let these flies out into the atmosphere, into the environment, and then they know where to lay eggs. They suddenly are able to kind of pick a plant out of this entire forest. And and then also like to get in a lot of fights. They're very aggressive. But it kind of blew my mind that they would be able to find somewhere to lay eggs just like that. So when I got the opportunity to do a PhD, I kind of was like, I think I'm interested in looking at, like what is inducing flies to lay eggs in this group. Let's see. Oh, you can watch this later. I'll show it to you again if you want. I got it on my phone. One of the big reasons too why I wanted to study this is that in my time in the lab, I've been trying to bring in species from the wild and rear them into colonies. And some of them just haven't worked. So this is a nice table of the amount of species that me and the state technician, Alex Zawori, who took my place, have tried to rear over the last 6 years. And, it's pretty good, but we also pick flies where, they're probably going to work out. So the fact that, out of these really good picks, only 58% of these species were able to be successfully reared in the lab says something. And there's also a historical precedent for being frustrated about flies not laying eggs in the lab. This is one of my favorite quotes that I came across in my reading from a really amazing dipterist, Forbes Robertson. And he said, it will be really pleasant to get back to Edinburgh and work with normal Drosophila, for these Hawaiian species literally make one psychotic because it's so difficult to get these guys to lay eggs in the lab and to rear them up. And of course, Dr. Robertson had his own troubles with that. So there's definitely a historical need for this research as well. There's quite a bit of history with other groups of Drosophila. As you might expect, having Drosophila melanogaster be a big genetic model and having a lot of invasive Drosophila be a problem too. There's a really big framework that I can work in when I'm thinking about my own Hawaiian Drosophila that haven't been studied yet, but have previously. So this is a great example of kind of the steps that most Drosophilas take in order to try and find some substrate to be able to lay in. So you have host location where, you're finding that tree in the woods. You have host acceptance where maybe you're tasting the rot on a branch or on a fruit. And then you have host use, which is laying, having the larvae kind of eat things and then fly away. So the thing that I'm really interested in for my work is kind of the olfaction and taste aspect of host acceptance. And this is because the flies that we have gotten to lay eggs in the lab do a great job laying in tissue that's soaked with host plant extract tea. So when I was trying to develop this research, that's kind of our first step on something we have to go on is that they're probably smelling something in that plant extract and they're probably able to taste something too. So that's kind of where I wanted to focus and center my research. And how do you do that? Perhaps you might be asking. Well, the first thing that we needed to do was collect some material from the wild. And this is really where I think the synergy between conservation and fly research and microbes come. Because something that's also really important about other Drosophilids is that they love to use yeast. That's a really common stimulant for flies. And so trying to find that out for Hawaiian Drosophila, whether or not that's an equally important stimulant is important. And so what we decided to do is team up with Dr. Hynson's lab, and Michael Fernandez and I flew in helicopters all over the island of Oahu, and we collected a bunch of plant material that you can see up here. Whoop, there we go. And then Michael was able to bring some of that into the lab, isolate some microbes from that, which you can see here, and make this beautiful microbial library that we now can use, I can use specifically, to try and test some of our fungal isolates on these flies to see if they're interested in laying in those. So the thing I'm going to tell you about today, the story, the tiny story I want to tell, has to do with these players here. I've done a lot of work over the last 2 years that it's too much. It's too much for 15 minutes. We all know. So I'm just going to tell you about these two experiments. So the players that we have that I'm going to try and see if I can make them lay eggs are Drosophila crucidra, which is a really common Oahu generalist. It will lay in pretty much anything. And then we have Drosophila netida, which is an Oahu specialist, which tends to lay only in CIDs. And so essentially what I did was I got colonies of these flies up. I got some tea from the branches that we collected and sterilized that so that there's no microbes living in that. And then I have some yeasts that we got from Dr. Hynson's microbe lab. And I'm essentially going to inoculate the sterile tea with these two yeasts. And I chose these because this ʻŌhiʻa is commonly found in fly guts, which means it's probably a good bet that these flies might like it. And then I picked this one because it is one of the only identified yeasts that we found in our CIDs extractions, which makes it a really good contender for maybe the specialists to be interested in. And then I designed some of these behavioral experiments where, one sec, so, oh my gosh, I'm so thirsty. Okay, thank you, sorry. Wow, that's better. All right. So the next step I had was this behavioral experiment that I wanted to set up. And essentially what I'm doing is I'm giving flies the option to pick different combinations of these teas and yeasts. And I wait for 72 hours and I see if they lay eggs. It's pretty simple. So you can see the boxes that I use. You can see the layout with the tubes. All of these tissues have some of that tea soaked in there. And then I have little pieces of agar that have the yeast cultures grown on. So over here, you can see an example of a tube that doesn't have anything, no egg laying whatsoever. And then a tube that does. It might be a little bit hard to see, but these things there are eggs. So I think that's all I wanted to say. Yes. Okay. Nope. There we go. Okay. So this is the first experiment that I did with that generalist fly, Drosophila crucidra. And these are very preliminary results, I should say. Like, Matt, I haven't gotten any models going yet. I tried some, but I didn't want to, so the thing that's really interesting here is that there weren't any significant results. The flies, these generalist flies laid in all three of my treatment groups and laid in these yeasts. Probably not preferentially, but I still have to check. But the interesting thing that I want to take home from this is that these non-significant results are actually really cool because the yeasts were able to kind of rescue the fact that there was water in these tubes. And so these generalist flies were interested in laying in the yeast, which is great. That's really interesting. And kind of points to the fact that these flies might indeed be finding some Ovi-Stimulants in these yeasts that I'm using. So that's really cool. There we go. Okay. All right. So now the specialist. This is Drosophila inedita, and it lays more or less just in CIDs branches. And you can see it's pretty dramatic results for which kind of tea it was interested in. My inedita didn't lay in water. They didn't lay in the Wickstromia tea, which is kind of an outgroup tea that I decided to use. And they quite liked the tea that their host plants in, which is like, I'm glad to get a signal for sure. The more interesting thing might be that after 72 hours, there was a slight preference for one of the yeasts, which was very cool. And it wasn't the one that is on Pisonia or on CIDs itself. Pisonia and CIDs are the same. I might have said Pisonia a few times. It's the same genus. Anyway, yes, so they were a little bit preferentially interested in this ʻŌhiʻa yeast, which was very cool to see. And might suggest that, yeast might bolster the effect of some of the tea metabolites that are kind of inducing these flies to lay eggs. So that was really interesting. All right. Okay, good. Conclusions and next steps. Generalists, I could kind of rescue that ovistimulant effect with yeast by itself, which was interesting and wonderful. And I think can probably lead us to some questions about what kinds of yeast, what kinds of metabolites these yeasts are producing, and kind of look more into what generalists prefer, or perhaps they don't. The specialist preferred the autoclaved host plant tea, but it seems as though those yeasts, either as a source of nutrition or as some sort of metabolite, were interested in preferentially laying for that. So the next step, you might ask, is what to do now. And the thing that I'm really interested in is maybe trying to isolate some of these different metabolites and these head volatile spaces so that if they are things that we can identify, we can buy them commercially and have flies come into the lab and we know exactly what's going to get them to lay. So we can eliminate that problem of frustratingly trying to bring flies in and not having a colony develop. So if we're able to identify some of those volatiles, then we're going to be able to conserve more of these species that are declining, and we're going to also be able to understand a lot more of their evolution, a lot more of all those interesting things that I talked about at the beginning. And I've done it. Okay. Thank you all so much for listening.
Hello, everyone. Thank you all for being here today. First and foremost, I'd like to thank the C-MAIKI team for setting this up today and the UH Foundation for giving me the opportunity to conduct this research as well as present it today. Today, I'll be talking about my first PhD thesis chapter, From Fire to Reef, Wildfire-Associated Shifts in Coral Microbiomes Following the Lahaina Fire. Before I give my talk today, I'd really like to take the time to acknowledge the community and the people within the West Maui area that gave me the opportunity to come in and conduct this research during such a vulnerable time. So first, to get started, I'd like to give a general context of what I'm going to be presenting today. Wildfires are increasing globally, with Hawaii having especially elevated risks. Following wet winters and Kona rain events, invasive grass create heavy fuel loads that dry by the end of summer, that increase wildfire risk across leeward Hawaii. This is especially important for a year like 2026, where we're projected to have a super El Nino with similar effects. On August 8th of 2023, the Lahaina wildfire created an unprecedented loss of life and property while becoming the first major urban wildfire directly adjacent to a coral reef ecosystem. In West Maui, the post-fire runoff creates a direct land relationship to the nearshore coral reef. This included transient mobilization of ash, sediments, nutrients, metals, and microbes from burned landscapes. Yet little is known how these kinds of transient and organic matter affect coral microbiomes. This is a wildly understudied area, and as I said, it's incredibly novel. However, these coral microbiomes can act as a sensitive bioindicator for environmental stress and ecosystem recovery. So I'm going to go into where did we actually study. We did coral sampling and used Hawaii Okava'iola's water quality monitoring in Honokauai, north of the burn zone, Kahekili, Wahakuli, Mala, and Olowalu. And it's especially important to note in this figure that there is a stream at the Wahikuli site, the Kahoma stream at Mala, and Olowalu stream down in Olowalu. So there are 6 major time points that we were able to study during this fire. The first being August 8th, when the fire actually happened. Now, on November 30th of 2023, that was considered our first sampling time point, our pre-first flush event. And this was the first time we were legally allowed to get into the water to collect the coral samples. The one major time point is going to be called our post-first flush. That was on January 18th, 2024. This is the first major rain event where a lot of that organic matter and material was actually put out onto the reef. And then luckily after that, we were able to do a 9-month post-fire study collection point, 12 months and 14 months. So we were able to kind of get this long-term data set of what was happening. So what were some of my hypotheses for looking at the coral microbiome after the fire? The post-first flush event will drive the strongest reef disturbance response. The 2 coral species in this project will differ in holobiont response to post-fire runoff. And lastly, coral microbiomes will show early disturbance responses followed by later recovery and or restructuring. So what exactly was collected? Coral biopsies were collected for microbiome and metabolome analyses. 8 samples were collected per species, per site, per time point. One thing that's incredibly novel about our approach for this is that our lab actually adopted, where are the pictures? There they are. There we go. Our lab actually adapted a prior method in the past from a alumni of Craig Nelson's lab, where we actually use a 16-gauge syringe tip to collect our samples. In the past, when people wanted to collect coral microbiome samples, they would often be the size of at least a quarter and use things like a hammer and chisel or a syringe to scrape it off to collect the tissue. ultimately leaving a large scar at areas for pathogens that could ultimately affect the coral health. Within this, you can actually see these two holes are the size of our biopsies, and within 2 weeks, they were completely healed within the coral. One thing that makes that incredibly useful for sampling like we do is that's smaller than a fish bite. So you're not taking these large chunks out when you're going to sample. It's less invasive than what's already happening on the reef, and we can still gain all of the positives from it. We also use the ʻŌhiʻa long-term water monitoring in West and South Maui. This is a group with DAR and TNC that's also volunteer-based. It's collected every 3 weeks since 2016, which gives us a very nice long-term spatial and temporal gradient, including temperature, salinity, turbidity, and various other nutrient data that's been processed at the S Lab on campus. And lastly, some rainfall data. So our two coral species are Paredes lobata and Montipora capitata. They're the most common across the Hawaiian Islands. Our Paredes lobata is a microbial regulator that maintains stability under stress, dominated by one group or one genus called Endozoicomonas, typically up to 90%. We also have Montipora capitata, which is a microbial conformer. So it reacts to the environmental stressors significantly more than our microbial regulator. This is going to be a common theme throughout the rest of my talk. So you can kind of think of Paredes lobata as being the hardier coral, whereas Montipora capitata is more reactive. And this is what it looks like if you were to go out and snorkel on the reefs. Two very different life history strategies in terms of microbial communities, as well as some of their growth patterns. So the first environmental context or environmental factor that I'm going to go over is our rainfall. And honestly, this is the one that really sets the standard for kind of how this talk is going to go. The rainfall actually occurred, the first major rainfall occurred in January 11th to January 18th as two major rain events. Prior to that, there wasn't any large rainstorm after the fire. So the mobilization of a lot of this organic matter and potential fire material didn't have the ability to move as freely out onto the reef. This is incredibly important that this rain event did not happen in, say, September or October before people were able to really try and manage it the best that they could. And it also gives context that allowed me to be able to use this collection point of what did the reef look like without some of these organic matter out on it. But being in Craig's lab, we do have to look at chemistry and water. So we had to look at an N to P ratio, which is very important for coral microbiomes. A high N to P ratio can lead to dysbiosis within the holobiont, which you can see in stress. Total N increased. All my pictures are delayed. Total N increased dramatically right after the fire, so the N to P ratio spiked. That's not very surprising considering there still was ash and a lot of the fire did go directly onto the beachfront or all the way up to the beachfront. And there is a lot of noise, but that's pretty on purpose for now. But you can see after the first major rain event, the N to P ratio drastically drops. That's from the nutrient loading of the first major flood or rain event. There's a large increase of N and P on the reef. And then we can also talk about ammonium, the bioavailable nitrogen. And why this is important is because of you have things like microbes, plankton, and algae and corals that all can uptake it. Increased after the fire in the first major rain event. So you can see these spikes at the fire, the spikes at the rain event, and then lastly, a little hint for what might come, but there's two sites, Waiʻikuli and Honokawai, that you can see it as well. So to get kind of back into this microbial conformer versus a regulator, we look at Paredes lobata. It maintains stability under stress, but there's less species richness. Just because there's less species richness doesn't mean that it's not as important or less stable. It just means that there's less players in the game. So when you do have some of them come and go, when you have stress, for instance, during our post-first rain event, that's a lot more significant than losing one for something like Montipora. Remember this Montipora, the conformer reacts far more to these environmental conditions and has increased species richness. So these are on the same scale. So even when you can see that some of the richness increases on the final time point and recovery restructuring, that's kind of just what it is in the Montipora in general, and significantly higher by the very end time point. So our microbial regulator, this is our Parietes again, it's dominated by this Endozoicomonas, this green group. You can see that it's, as I said, can be up to 90% of the entire relative abundance. And then you'll see this pink group, this Vibrio, this opportunistic copiotroph, increase under stressed events for Parietes. For instance, this event, these 3 bars right here, are all of those 3 sites that actually had the streams when we looked at the map. The other 2 you can see look virtually unchanged, and they just don't have that major flush or major runoff event. And then my best guess is why we see these 2 large spikes here was that point that I made during the ammonia, that bioavailable nitrogen becomes available that causes spikes in these copiotrophs, which is nice. And it's this hardy coral species, it takes a lot to kind of change their microbiome. And something like that, I think, is very important in the context versus Montipora capitata, because you need like a real event to actually do something. So if you're monitoring this over the course of space and time, you'll actually be able to say that something happened on that reef, not guessing. There's a rain cloud for the event. And then we have Montipora capitata, which is the complete opposite, this conformer. You can see that it's no longer dominated by this green Endozoicomonas group, and in fact has a much larger pink Vibrio group that we just saw on the slide before. However, the difference is you can see that there are a lot more groups in this first time point and in this final time point. The Vibrio, as you can see, was dominant across all sites, even the sites without the stream. That's because this species is far more flexible and far more fluid with some of these environmental stressors. And then by the time you get to time point 4, you do start to get this green Endozoicomonas healthy microbe back for these corals and a little bit of restructuring. And then eventually, by the final time point, you do see this increase in diversity, which is normal and cited in literature. So for my entire conceptual model of this, there was a fire. It's hard to say that at any point that Lahaina was lucky, but they were lucky that there wasn't a first flush anytime immediately after the fire, that there is able to have some mitigation to dampen some of this to go onto the reef. This did eventually go to the reef, changing the water chemistry, altering some of the coral microbiome. What's incredibly interesting about my project is that people around the world don't study coral microbes in the terms of long-term ecological monitoring. When you're studying coral coral ecological monitoring, you look at things like presence absence, shifts in growth, overall changes in reef. You say there was a bleaching event and you lost all of this, but you don't look at the fine scale of, are there stress indicators before then? Can you look at microbes? Dr. Gilbert, I think, did a really nice job of basically saying, how do we get people to care about coral microbes? And I think that for me is really important. You can use this as an ecological tool to start saying that certain sites have certain stress. And when you have places like ʻŌhiʻa that have done this long-term monitoring, since 2016 and almost 10 years of data, you can start to really contextualize what you're seeing for the future. So with that, I'd like to say thank you again to the people of West Maui, my fiance Claire for going out to collect every coral with me throughout the whole time, the rest of our labs and teams that you can see here, and all of the different organizations, including the UH Foundation for supporting this research. Questions?
Thank you, Nicole. Aloha mai kako. My name is Sebastian Church. I'm a 5th year, going on 8-year PhD student in tropical plant soil sciences. Kiana Frank is my PI. Shout out Kiana Frank. What's up? Welcome back. We love you. Let's see. I'm born and raised in Miami, Florida. I've been living in Makiki for about 5 years now, and I perform research at Kewalo Marine Laboratory. The talk I'm sharing today is more of a story. It's about how a side quest has become a main quest and likely another chapter of my dissertation. So this is a cautionary tale to PIs. If you take leave and leave your students to their own devices, which Kiana already knew this was happening. She already knew this was going to happen. So to be fair, enough about me. Let's talk about There we go. Let's talk about Adam. Adam is a taro farmer in Keikaha on the west side of Kauai, and Adam can grow some mean taro. That is oha. If you know taro, there's makua, the center corm, and then there's oha, which is like the keiki. That's oha. That's massive for oha. So this is from the first harvest at this site in Keikaha. But subsequent harvests have all crashed before maturity. So what Adam's observing is about 4 to 5 months. There's dieback in the upper parts of the plant. And when he pulls the corms, he sees this, let's see, oh, there we go. He sees this soft rot, very defined with this horizontal line. And then this corm up top is actually harvestable. So he's been able to recover some, but his yields are down like to 25, 30%. And where there isn't that soft rot, there's this hard rot that colloquially is called guava seed. So Adam hit me up back in 2023 about this project. I met him originally through one of my earlier dissertation chapters. And so shout out to a couple of people here. One is a crop consultant here in Hawaii. Dr. Jonathan Dienick is a professor in tropical plant and soil science. And then Roy Yamakawa is a retired extension agent who worked with a lot of taro growers in Hanalei. And you can see 3 legends, 3 different ideas, right? And then you bring in Adam, who's the farmer and an expert in his own right, and he has a 4th hypothesis. Since Adam's the farmer, we go with Adam's idea. So we set up an experimental trial, a field trial in one of the patches in Keikaha. We just did different amounts of calcium additions. It was an awesome day. A couple of ohana from Keikaha came out and helped put the experiment together. So just real quick, mahalo to the Vidinia ohana and the Kaiahue ohana. Thank you guys. It was awesome to jam together. And then of course Nihi holding it down. And so outcomes from that experiment were that calcium really didn't do much. We measured the recoverable corm because that's what's relevant to Adam. That's what he can sell. And we didn't really see a whole lot of difference across the experimental plot, nor across the entire 8-acre farm. So this sort of like this condition with the soft rot at the bottom, kind of like rotted out from underneath, is present in every patch. So we wanted to, let's see, we didn't get like much conclusive or direct solutions from that experiment, but it did allow us to kilo extensively. And so one thing we noticed was wherever we were finding the hard rot preceding the soft rot in the corm. So taro grows from the bottom up, and we were noticing that the hard rot is always preceding the soft rot. Still yet, it's not necessarily linear. So, So just in not all corms were the soft rot and the hard rot adjacent. And we noticed that the hard rot was actually a little bit more associated with like newer flushes of roots. So the soft rot occurs, all the roots die out. And then where there are new flushes of roots, that's where we were seeing the hard rot. Another tricky thing about this investigation is we weren't seeing a lot of foliar symptoms, which is usually typical of plant toxicities or deficiencies. So we decided to investigate further, run samples on soil, leaf tissue, corm tissue, and we actually extracted the guava seed nodules out of the corms and ran that as well. And so soil was low in potassium and calcium, high in magnesium, iron, manganese, and sodium. Leaf tissue was similar to soil, except manganese was low instead of high. Comparing healthy corm to affected corm, manganese and sodium were both higher. And then in the guava seed nodules themselves, manganese was an order of magnitude higher in those guava seed nodules compared to healthy corm. So we kind of just laid out all of these, talking to Adam, just chopping it up and trying to figure out what's going on. And so we came up with these abiotic hypotheses, thinking, okay, maybe it's manganese, which is very common in flooded soils, manganese toxicity, due to reduction oxidation reactions. And then we also thought, okay, maybe there's some kind of salt or sodic situation happening, an accumulation of not just sodium, but ammonium, potassium, magnesium as well, because these soils are very high in magnesium. So we kind of got to this, and then I went back to professors. Ty, thank you for your guidance. And I talked to Ty. Ty said, well, if you really think it's abiotic, you got to eliminate the biotic causes. And so the main biotic agents in wetland taro are oomycetes, or these pseudofungi. They're the main agents that cause the soft rots, namely Pythium, Phytophthora, Phytopythium, Globisporangium. So we took corms from the patches in Keikaha, sent them to 2 separate labs, Dr. Marion Luis's Mycology and Plant Disease Management lab, and then Dr. Josiah Marquez's lab. They extracted corm tissue with the guava seed, plated it, and then extracted DNA from the isolates and ran that through Sanger sequencing to identify them. And it turns out they both found the same thing, Phytopythium helicoides. And so this is one of those oomycetes. It's it's very good at proliferating in water. The zoospores, which are like the seeds for the pseudofungi, they're motile. They can swim, so it spreads very readily. Another relevant aspect is that Phytopythium helicoides has never been associated as a disease or described as a disease of taro in Hawaii. So if we can build that association, this would constitute a first report. So our next step in order to investigate that association is to run a pathogenicity trial. It's very basic, uninoculated treatment, and an inoculated treatment where we insert a plug of Phytopythium helicoides into the corm, let it grow for a little bit, and see what see if there are any symptoms. So that's coming up. Now, if you were here when I pitched the whole mahi microbes idea, you would be sitting here like, Sebastian, that is not what you said you were going to do. That has nothing to do with it. And you're right. It didn't have anything to do with it until we stumbled on Phytopythium helicoides. So the original idea with this project that I pitched was I would verify and develop qPCR primers that could identify and quantify pathogens in soil. That was the original idea. So now we're just adding Phytopythium helicoides to the list of pathogens. Sweet. Okay. qPCR basically and primers, just a real quick crash course. This is DNA, or like genetic material, right? So these lines here, this orange line and this black line, genetic material. The black line is the DNA of, let's say, the pathogen or the organism that's in the soil. And then the primer is a piece of genetic material that binds to the DNA from the sample. And when it binds to that DNA, it traps these fluorescing compounds. And then that's how these machines count how much of that genetic material is in the sample. And then that gives us an estimate for how much that pathogen is occurring in the sample. So it's a quantitative method, which is pretty cool. But you need the primers, and the primers are specific to different species. So every species needs its own primers. So I've been working on that. These are just some species. So you can see Phytophthora, Pythium, Globisporangium, and then there's Phytopythium helicoides as well. So I've been working on verifying these, running them through some software, doing some in silico stuff. So Amplifex is an awesome software, and then blasting stuff, which is fun to say. So once we get those numbers, right, quantify pathogens, we can then take that back to growers. They can make management decisions, whether to delay their planting, drought their soil longer. What else can they do? Intercrop, bring in other crops, fallow longer, all sorts of different stuff. Just don't put chemicals in the lawn. And so, yeah, this is Adam's farm. So back to Adam. Adam is super persistent. And he's a super determined person. You have to be if you want to grow taro in Hawaii. So big up Adam. We started another trial in January of this year, applying wollastonite or calcium silicate. There's a long history of using silicate in soils in Hawaii to improve plant growth. So Adam reads voraciously. I didn't realize I would have so much homework with this project, but he just texts me papers, and I'm like, dude, you don't even have an account. Like, how do you get access to this stuff? And yeah, so we're trying one off tonight. So we're carrying on. So yeah, but I want to say thank you to all these people. All these people. Adam, especially thank you for being a mentor, a friend, and a teacher in spite of the homework. All these guys from Kekaha. This is Brother Kelsey, took the drone shots. Kiana for trusting me to be by myself. All of these guys, they help Adam make poi Mondays and Fridays in Kapa'a, and without their support, Adam won't have time to do investigations like this. So they're critical to this whole thing. Jonathan and Ty, thank you guys for all of your support, all the conversation and insight. Dr. Luis and Josiah, you guys are just getting to know me. I'm so sorry. No, I'm kidding. I'm so stoked to work with you guys. And thank you for being so open and willing to collaborate. This is the elusive Pete Bunn. I've talked to Pete so many times over the phone and through email, but I've never seen him in person. So I'm kind of wondering if he isn't like Jonathan's other, like, what do you call it? His like secret identity or whatever. But Pete's awesome. So Pete, thank you for all the wisdom and insight and experience. So generous with his knowledge. And then this is Catherine Wrong with WRC Environmental Chemistry Lab, who ran the ICP samples for us. There's a lot more people to thank. I also want to thank you all for your interest and attention. People online, I see you. Mahalo. Thank you.
Okay, good afternoon, everyone. My name is Stephanie Murray, and I'm a computer science PhD student at UH Manoa. I work in the Pacific Biosciences Research Center in Pipes Lab, and we are doing computational biology. And I'm here today to present our study on identifying the protective microbiome of rapid ʻŌhiʻa death survivors. This study came about as one of several ideas in our lab when we wanted to do something different to our usual projects and work on something that benefited the Hawaiian ecology. I presented this project to become one of the winners of the C-MAIKI Michael Bruno Award for Excellence in Microbiome Science. And this award is supporting this project. At the beginning, I made contact with people from different organizations working on rapid ʻŌhiʻa death. And I was led to Mark Hughes at the USDA Forest Service, who also works with the Akaka Foundation ʻŌhiʻa Disease Resistance Program, who we are in collaboration with for the study. We are currently in our beginning stages. So today I'm just going to briefly cover some background on rapid ʻŌhiʻa death. Then I'll go into our research question, hypothesis, and study design. ʻŌhiʻa trees are the heart of Hawaii's native forest. ʻŌhiʻa supports native biodiversity, stabilizes soils, regulates watersheds, and sustains cultural practices woven through the Hawaiian identity. Its loss will alter forest structure, species composition, and the health of island ecosystems. With no cure available, even a small clue about why some trees survive rapid ʻŌhiʻa death could change everything. So rapid ʻŌhiʻa death, or ROD, emerged in 2010 on private properties, and it was officially confirmed in 2014. ROD is driven primarily by two aggressive fungal pathogens, Ceratocystis leuco-ʻŌhiʻa and Ceratocystis huli-ʻŌhiʻa. These pathogens attack the water flow of the trees. And it is found on the islands of Hawaii, Maui, Kauai, and Oahu. Since its detection in 2010, ROD has resulted in millions of ʻŌhiʻa deaths across the islands. It has affected more than 10% of ʻŌhiʻa. Understanding pathogen biology and spread mechanisms is crucial for developing effective management strategies. So, how do ʻŌhiʻa become infected? ROD begins when fungal spores infect a tree through wounds in the bark, and this causes rapid browning and it can lead to death. The spores hitchhike on sawdust and other tree fragments, and once they reach a forest, it really can't be controlled very well. And these images on the right show what ROD looks like when it's presenting in tree trunks. So our key question here is, why do some trees survive when others perish under similar conditions? We think that specific microbial communities associated with resistant ʻŌhiʻa clones confer protective effects. Investigating the sapwood and soil microbiomes could reveal candidate bacterial and fungal taxa promoting resistance, offering novel biocontrol opportunities. So now I'm going to talk about our study design. After doing research and discussing with experts on ʻŌhiʻa, we settled on a clone-based in-greenhouse design. In nature, there are too many variables that we cannot control. We don't know if a tree is resistant or just lucky, and the microbial communities on dying trees might be different simply because they are in a different state. So what we're trying to determine here are, do clones that differ in ROD severity also differ in their baseline microbiomes? So here is a diagram of the clone-based study. We take clones that have already been classified as resistant or susceptible. We grow them under controlled conditions and compare their baseline microbiomes before inoculating and collecting samples at a later stage. Mark Hughes and the Akaka Foundation have been running trials on different genotypes in their greenhouse. They have ranked them by wilt severity, and we are going to focus on extreme phenotypes, baseline sampling, and control conditions to isolate the biological differences. So now we come to selecting the clones. Here is a graph produced by Mark Hughes and the Akaka Foundation on one of their previous screening trials. They are ranking the wilt severity between 0 to 4, as we can see in this table here. And what we would be looking for is selecting 2 from the most susceptible categories, and then 2 that have not shown any wilt, making them resistant. Here we have an, nope, we do not have an image. This is our sampling strategy. We are going to sample sap, wood, and soil. And for our design, we will use 4 clones, 2 treatments, and we are going to have a pathogen treatment and a control, and 6 replicates per condition. Okay. So now moving on to our inoculation progress. We have an image from the ʻŌhiʻa disease resistance program showing how inoculation is performed. So plants will be inoculated using a standardized stem wounding protocol, and the wounds in the trees will be treated with either the pathogen Ceratocystis leuco-ʻŌhiʻa or blank filter paper, which is the control. The plants will then be monitored for symptom development following inoculation. So we are sampling, like I said earlier, sapwood and soil, and we will be taking samples at different points So for the susceptible and the matching controls, those will be sampled at between 4 to 8 weeks. And then we will have late stage sampling of the resistant and their matching controls. And those will be harvested either at the severe wilt level 4, or if they were not infected, we will be harvesting them at the trial completion at 6 months. So we're going to use 16S and ITS sequencing to profile bacterial and fungal communities with controls included. And these methods enable high resolution identification of taxa with potential roles in resistance or susceptibility. For analysis, we are going to compare microbial communities between groups using diversity metrics and identify taxa that are associated with resistance. We are looking for candidate taxa that are low in abundance in the resistant clones at the baseline and high in resistant clones following the inoculation. So in conclusion, the study tests whether microbiome differences are associated with resistance and identifies candidate taxa for future work. We have the potential to provide new hope for Hawaii's forests. And I would like to say thank you to Dr. Lenore Pipes, who has been helping me come up with the study design, as well as Alexandra. And of course, Mark Hughes has been an integral part of this process, and the Akaka Foundation for allowing us to use their facilities. And of course, C-MAIKI and the Michael Bruno Award. Thank you very much for your support.
Aloha mai kākou. My name is Mamo, and I'm a PhD student in the Medeiros Lab, Hui, led by Dr. Matthew Medeiros. So as a Kanaka growing up on the continent, I used to think that my biggest challenge to understanding ʻIke Hawaiʻi was the physical distance between me and Hawaiʻi. However, through living here, I've learned that actually a lot of Kanaka who were raised in Hawaiʻi have at times struggled with their Hawaiian identity as well. And our shared source of disconnection is because Hawaiʻi today is politically and ecologically distant from what generations of our kupuna, our ancestors, experienced. This featherwork in the Bishop Museum, crafted with extinct manu like o'o, o'u, and the mamo, is a reminder of that. And of over 50 species of Hawaiian honeycreeper that once existed, all but 17 have gone extinct, and 11 of them are endangered. So I believe the only way to truly know the manu or our birds is, so the only way to know our manu, the birds, the relationships that they have to the forest, as well as the relationship that our kupuna had with them, is to spend time with the species that are still alive and to develop our own relationships with them. And it is our kuleana to ensure that future generations will have that opportunity too. So my research is focused on the Culex quinquefasciatus mosquito because the diseases they spread, like avian malaria, are one of the main drivers of manu extinctions today. Currently, there are efforts to lower the mosquito population by releasing lab-reared male mosquitoes, which carry a bacteria called Wolbachia that acts as a birth control. When these males mate with wild females, the eggs that they produce do not hatch. Wolbachia is one of the most famous examples of a bacteria within the mosquito microbiome that affects reproduction. However, mosquitoes also typically have about 200 other bacteria in their microbiomes, like this Aeromonas species, which can also affect nutrition, development, and reproduction of the mosquito, which in turn affects the mosquito's ability to spread disease. Therefore, studying the microbiome of mosquitoes is necessary for understanding mosquito biology and important for ongoing and future mosquito and disease control efforts for avian conservation in Hawaii. So in order to start identifying microbes that are relevant to mosquito health and fitness and functioning, I conducted a natural experiment at Lion Arboretum to survey wild Culex mosquito microbiomes. The Medeiros Lab helped me set up 18 of these plastic totes, which were filled with water and the leaves of kukui and hau plants. in order to mimic natural mosquito breeding habitats. And so once these toads were colonized by Culex mosquitoes, I collected samples from all their different life stages, egg rafts, larvae, pupae, and adults, as well as water samples. Back at the lab, I dissected out the midgut and the malphigian tubules of the mosquito samples and sequenced the DNA libraries of all sample types using the Earth microbiome 16S primers, targeting the 16S gene of bacteria and archaea. I processed the sequences using the data2 and Lulu packages in R, and also conducted the statistical analyses in R using phyloseq and GLMM-TMB. So this is an NMDS plot of my samples, which suggested that the bacteriomes, or the bacterial portion of the microbiome of my sample types, generally clustered together, suggesting that they had more similar microbiomes. And so to tell if this pattern from the NMDS was real, I ran generalized linear mixed models, or GLMMs, to test my hypotheses about how mosquito life stage and tissue shape bacteriome structure. So these are my three hypotheses. The first one was that the bacteriomes would vary by sample type. So this data set included all of my samples, water, rafts, larvae, pupae, and adults. And the best fit model for my group of models supported this hypothesis. My second hypothesis was that the bacteriomes would vary by life stage of the mosquito, but that variation would also be dependent on the tissue that I dissected. So this included larvae, pupae, and adult samples. And my best fit model also supported this hypothesis. The third hypothesis that I tested with the GLMMs was that the bacteriomes would vary by life stage, tissue, and sex. And the best fit model only included life stage. So in that case, my hypothesis was not supported. So now that I knew that these different sample types and tissues had different bacteriomes, the next question was, do these different bacteriomes actually have different functions? So with support from C-MAIKI, I conducted metagenomic sequencing on a subset of 48 samples from my original sampling to study the changes in functional gene potential across life stage. And I processed this data through a bioinformatics pipeline called Squeeze Meta, and then use phyloseq and GLMMs again for my analysis. So I just got this data back really recently, and yeah, the following slides are pretty preliminary. So in this NMDS, you can tell it looks really different from the one from the 16S data, but the biggest things that I'm noticing are that the dispersion for my egg samples, the adult mosquitoes and the pupae, the spread in that data, or the dispersion, is a lot larger than my larvae and egg or water samples. So I'm not really sure why this might be, but we're gonna look at the GLMs. So I ran models to test the same hypotheses that I tested on my 16S data. The first one was that the bacteriome functions would vary by sample type, and my best fit model did support that. The second hypothesis was that the bacteriome functions would vary by life stage and tissue. In this case, the best fit model said that bacterial functions vary by life stage, but not by tissue. And then for my third hypothesis that I tested on my 16S data, I wasn't actually able to test it on my metagenomics data because I only sequenced female samples. Oops. So this is a tree chart representing the metabolism genes with the largest estimates from my best fit model associated with hypothesis 1, which was the sample type model. And each colored box represents a different type of metabolism, and the size of each box is proportional to the number of genes of that type. So on the left is a plot for larvae, and on the right is a plot for adults. And overall, larvae had more bacterial genes assigned to metabolism than adults. I'm not really sure what this means, but if I had to guess, the bacteriome of larvae might have, it might mean that the bacteriome of larvae just have greater functional diversity, or that the microbiome or the bacteriome of adults is more deterministic. There we go. So this is just like a really preliminary look at some of the categories, but if we zero in, or if we look at energy metabolism, larvae had 2 genes assigned to methane metabolism, or the breaking down of methane, whereas adults didn't have any. And I'm not exactly sure why this might be. I wasn't able to find in the literature anything about methane metabolism in mosquitoes. But I do know that the larvae gut, for instance, is an alkaline environment, whereas the gut of mosquitoes is usually acidic. And when I was looking at the literature, there are several papers about the digestion of wastewater sludge that reported that they were able to collect more methane from alkaline fermentation than acidic fermentation methods. So this is kind of a stretch, but maybe the alkaline environment of the larval gut is supporting archaea that are generating methane. And so in turn, there needs to be a bacterial community that can metabolize that. Another comparison I want to touch on was genes for furfural degradation, which were again enriched in the larvae samples. Furfural is a compound derived from the breakdown of plant material, and mosquito larvae feed on plant material. So it makes sense that they would have a bacterial community with this gene function. On the other hand, adult mosquitoes are almost exclusively drinking nectar, except for when they're occasionally drinking blood. So it would make sense that they do not necessarily need or rely as much on a microbiome that has the ability to break down furfural. So in conclusion, the results of my study show that metamorphosis, or the changing of life stages, really shapes the microbiome significantly. That bacterial members and their functions differ between the life stages, which suggests that different life stages require different functions from their microbiome. And so my future work will focus on what those differences in gene function are, especially between the larvae and adults, and also the water and larvae samples, and what those differences mean in the context of mosquito biology and mosquito control for avian conservation in Hawaii. So just a huge mahalo nui to the funders of this project, C-MAIKI, the Melanie G. F.G. Bros Fellowship through the UH Manoa EECB program and the NSF-GRF. Mahalo to Dr. Don Drake and Liloa Dunn and the Lion Arboretum for the opportunity to conduct this research there. And thank you to Nicole Yonoishi and Dr. Alexa Berger of MGAL for their help with the library preparation. Dr. Jenny Saito of the ASGPB for sequencing the 16S data, and Dr. Carolina Peplowska of the UH Cancer Center for running the metagenomic sequencing. Mahalo to my PI, Dr. Matthew Medeiros, as well as the whole Medeiros lab hui, past and present, especially Dr. Nicola Creefield and Dr. Chase Griffin for their help with the bioinformatics, and Ivis and Kellen for their help in the field. Thank you to Charisse of the Kelli lab for helping me with getting started on the COA HPC, and mahalo to my committee and everybody in the audience.